Seem Kavar. I am a pediatric gastroenterologist here at Stanford Children's Hospital, affiliated with Stanford University. It is my great pleasure to introduce to you Dr. Nielsen Fernandez Becker. She and I have not only been friends for many years and colleagues for many years, but as you'll soon hear, she's an incredibly qualified and inspirational speaker in the area of celiac disease and specifically breakthroughs, which she'll be talking about today. She received her bachelor's of science in biology back in 1994. Went on eventually to do an MD PhD at the Albert Einstein College of Medicine, completed her internal medicine residency and then gastroenterology and hepatology fellowship in Boston, initially at Harvard Medical School, and then at Beth Israel Deaconess Medical Center, also affiliated with Harvard Medical School. She is currently an adult gastroenterologist here at Stanford University, a clinical associate professor who has a lot of research experience as well as clinical experience in celiac. She's also currently the medical director and clinical director for Adult Celiac Center here at Stanford. So it's with great pleasure that I introduce Nielsen.
Thank you so much for that generous introduction. I'm going to share my screen. Okay. Here we go. Do you see my screen okay?
Yeah.
Great. Okay, so I was asked to talk about celiac breakthroughs and latest clinical trials. There's a lot going on, so I had to be a little selective. This is the outline for my talk. We'll start with celiac disease overview. You may have heard this before, so bear with me, but I think it will be helpful to understand what are the areas where there are questions and there's active research, but also it will lay the foundation to understand um, the what the rationale for some of the newer non-dietary potential non-dietary therapies are. So why do people avoid gluten? When we think about gluten-related disorders, they come in three broad categories. Autoimmune, which is celiac disease, but there's also allergic and non-autoimmune and not allergic, which is non-celiac gluten sensitivity. And only in celiac disease do we see mucosal injury, and we also understand the mechanism a little bit better than the other two. So gluten is basically a storage protein in wheat, barley, and rye, and it is a big bulky molecule that has a lot of prolines and glutamines, and it's broken down so far in the intestinal border, but there peptides or little pieces of the protein that remain intact and are capable of activating the immune response. And we're going to talk a little bit more how that happens. And it is the high proline contact of this that makes it resistant to the brush border enzymes. So we humans aren't well equipped to completely break down gluten, which is part of the issue. So big picture, we have a dietary protein that when ingested, causes an inappropriate immune response, and that leads to injury and destruction of the lining of the small intestine. It used to be thought that celiac disease was a rare disorder, but that in fact it affects about 1% of the world population. So that's a lot of people. And there are some areas on the map, like in the areas where it's gray, where we really have very little information about. So it might be even more. And just to orient you to what's happening in celiac disease, so it primarily affects the small intestine. And the small intestine is a vital part of the intestinal tract. You could live without a stomach, you could live without a colon, you cannot live without a small intestine because it's where you absorb most nutrients. It is about 22 feet long, 20 to 20 feet, 2 feet long. And the lining is organized, it's folded up in these small finger-like projections called villi, which increase the surface area. And celiac disease basically causes destruction of those villi. This is what it would look like, just a magnification of that. And under the microscope, this is what we would see. And here, basically, the villi are the little peaks, and the valleys are what we call the crypt. And that's where the cells that will then grow up to be cells that make up the villi are. And what I want to point out is that the intestine is actually a pretty amazing organ. What you have is like a single cell layer, and that's what separates everything that we eat and we ingest from what's right beneath it, which is the immune system. And it's an interaction between what's happening in the lumen or the inside of the intestine with the immune system right below it that is at the crux of the problem here. So this is a very simplified chronology of what happens in celiac disease. So what you have is that ingestion of a dietary protein, this time gluten, in genetically predisposed individuals causes, in a subset of genetically predisposed individuals, causes an inappropriate immune cell, an inappropriate immune response. That leads to inflammation. That also leads to T cell activation, activation of anti-TG2 autoantibody, which we're going to discuss a little bit more so you'll know what that means. And it's this misguided attempt of your immune system, or what I call friendly fire, that results in injury and destruction of the lining of the small intestine. And what you have is that you go from a very delicate, fluffy small intestine, as you see here, and right below is what you would see under the microscope, very tall, tapery villi, and very compact crypts, which is what we call the valleys. And over time, the villi are destroyed, so what you get is like a very smooth lining that's incapable of really absorbing nutrients in a normal way. And the microscope, what you see is that the villi are basically destroyed, and there's a lot of inflammatory cells in that immune compartment that we call the lamina propria. And this figure is one of the old ones, but I love it because it kind of gives you a snapshot of what's happening. So this is a figure depicting that single cell layer that I showed you before. And the green rectangle here is gluten. So what happens is gluten somehow gets through those cells and into the compartment where the immune cells are. There's a protein that's called tissue transglutaminase, which lives there. But in patients with celiac disease, what happens is that protein will then modify gluten in a way that it makes it more toxic, basically. It has higher affinity for the immune system, and that leads to activation of immune cells that are called T cells, and those in turn can cause injury to the lining directly by causing cell death. But they also produce a lot of chemicals or cytokines that will then increase the permeability, will make the gut a little bit more leaky. That allows more gluten to get into that compartment, and it basically amplifies that. It becomes a vicious cycle. At the same time, cells that make antibodies are activated, and those cells will make antibodies to that protein, tissue transglutaminase, which shouldn't happen because that's your own body's protein. And that's the hallmark of autoimmunity, that you are making an antibody against your own protein. But also, for reasons that we don't understand, the body also makes antibodies against the toxic component of gluten, which is gliadin, and specifically deaminated gliadin, which is the form of gluten that is created by tissue transglutaminase. And the reason I'm telling all of this is because those antibodies are what we use to make the diagnosis of celiac disease. And they're not thought to contribute to the injury, but they're markers that your body is actually reacting to gluten. And this cycle repeats itself over and over again when somebody with celiac disease eats gluten, and over time, those villi get shorter and shorter and shorter. And again, we get that transformation of the nice lush villi to the more smooth snakeskin-like lining. So genetics is a big part of this, and I'm going to go over that a little bit. So it turns out that 99.9% of patients with celiac disease carry one of two genes, which are called HLA-DQ2 and DQ8. When you look at the general population, about 40% carry this, but it's only a small subset in the order of 3 to 5% that actually develop celiac disease. And a big question in the field is, how does a genetically predisposed individual develop celiac disease? And the short answer is we don't know. There is mounting evidence that perhaps anything that causes enough injury to the lining of the small intestine, like infection with a virus or food poisoning, that allows gluten to come in contact with the immune cells in the first place, might be what sets it in motion. But we are still working on that. This is really helpful in patients who are already on a gluten-free diet, which you will hear from me is one of the challenges that we have, because we don't have great tools to make an accurate diagnosis in that scenario. But checking these genes is important because and helpful, because if they're not present, it essentially rules out celiac disease as a possibility. So it can be a helpful tool. Okay. So whenever there's a clinical suspicion for celiac disease, we turn to screening antibodies. And those are the antibodies to tissue transglutaminase and deaminated gliadin. And each organ system has its own type of antibody. In the gut is IgA, and that's why we check those. There is a small subset of patients with celiac disease where IgA is low because they have IgA deficiency, and then we have to rely on the IgG counterparts. But the bottom line is that the IgA variety of these antibodies are highly sensitive, meaning that they pick up the disease most of the time, in the order of 95 to 98% of the time. And they're also highly specific, meaning that when they are positive, it truly picks up celiac disease. There are very few false positives. Not perfect, but very good for a screening test. They need to be done while the patient is eating gluten, because what happens is that those antibodies go away and then we lose our opportunity to make the accurate diagnosis. Once the antibodies are positive, the next step is to document whether there is mucosal injury. And there we do an endoscopy with biopsies of the small intestine. And I'm showing you that same figure again. This is what we're looking for grossly, whether there's a presence, gross appearance of injury. And under the microscope, there's ways also that you quantitate how much injury there is. Also should be done on a gluten-containing diet, because once gluten is taken out of the equation, the lining heals, and it might be difficult to make the diagnosis. And it is an invasive test, so there's a impetus to try to develop better diagnostic tests. And I'm going to give you some examples of what's going on right now, that someday the biopsy may be replaced. But currently, it remains the gold standard to make the diagnosis of celiac disease in adults. In kids, there's a non-biopsy pathway that was started in Europe, but in the United States is still a case by case. It depends on how high the celiac antibodies are. I'm not going to spend too much time on this, but basically there is an alternate pathway for kids. This I'm showing you, this is a diagnostic algorithm, but the real point of it is to show you that it can get complicated, particularly because a lot of patients, not for their fault, but they're advised to start on a gluten free diet immediately without the diagnosis when the diagnosis is not well established. And then we have to, sometimes the gold standard is to reintroduce gluten in order for us to make an accurate diagnosis. And I know exactly how it sounds like. If somebody has celiac disease, we tell them, gluten is your poison. But then I'm telling them, eat gluten so I could tell you that you have celiac disease. So it is something that, from a research perspective, is being studied very significantly because we really do need better tools to be able to make an accurate diagnosis in an individual who's already on a gluten-free diet, and you'll hear more about that. But sometimes there's IgA deficiency. There are also other conditions that can give you the same exact gross look of what celiac disease looks like, but it's not celiac, and the antibodies are not made. And so it can get very complicated. And the tools that we have are pretty good, but they're not entirely specific to make the diagnosis. And as such, we need better tools, and we'll talk about what those are. The treatment right now, the only treatment is removing gluten from the diet, which, as you know, is easier said than done because gluten is in a lot of things that we wouldn't normally think of. And there's a learning curve, and it's something that has to be maintained. And even when we're as conscientious as you possibly can be, there are times when there are accidental exposures. And we cannot tell whether I mean, basically, we don't know that there's ever a not harmful dose of gluten. The thought is that in order to continue the inflammation that's required to cause a mucosal injury, that requires repetitive exposures. And it takes as little as 50 milligrams of gluten, which is not a lot. But the reality is that we don't have tools that are sensitive enough to pick up what the effect of a single exposure is. And so therefore, we try very hard to make sure that there are no exposures because, as I said, there is no known safe dose of gluten. And it's also really important, to keep on looking for other alternative therapies because a gluten-free diet isn't effective for everybody. And this is another one of those algorithms that's very busy, but the idea is that how do we follow a patient after they're diagnosed with celiac disease? We have some guidelines, but they're imperfect, and we don't have great tools to really tell if someone is adequately gluten-free. The antibodies, great screening tests, but they may remain positive for a very long time, depending on how high they were to begin with. Usually we say they'll normalize in 6 months to a year, but it could be later. And they don't really correlate very well with the health of the small intestine. And we don't have great tools to really tell how well healed the lining is. And so we tend to recommend biopsy after a certain period of time, which is usually 2 to 3 years. However, um, there's not great standardization in that process, and there's some subjectivity. So sometimes it's not very helpful, and it's very hard to tell how much improvement there has been. And the other thing is that at least in the adult population, significant number in the order of 40% based on clinical trial data um, adults will continue to show some degree of mucosal injury, even if these people are completely asymptomatic and the antibodies are negative. And it's hard to understand really what the significance of that is. So again, we need better tools. Okay. Okay. So, in summary, celiac disease is an autoimmune disorder caused by an inappropriate immune response to gluten. And the diagnosis requires there to be a clinical suspicion. We have the antibodies as the tool. We have the endoscopy with biopsy, but the diagnosis can be complicated. And the treatment does require lifelong gluten diet adherence. And again, the gluten-free diet is not effective for everybody for a variety of reasons. So the research that we're doing is critical to develop more tools to diagnose, monitor the disease, and to develop more new therapies. And now I'm going to shift gears to discuss some of that research. So I'm going to share with you some research that our group has done here at Stanford. One of those was that we developed a human model for studying celiac disease. So scientists use many different tools to study human disease. That includes animal models, and that includes cell culture models. And animal models aren't exactly small humans. There's significant differences. So it's always important to try to understand what's happening in the humans, and this is what we attempted to do. And some of you in the audience may have contributed to this, so thank you. So basically, my celiac patients and also controls uh, donated some biopsies. And my collaborators, this was led by Dr. Calvin Kuo, and the experiments were done by a very talented postdoctoral fellow by the name of Antonio Santos. So basically, they develop something called an organoid, which basically has all of the components of the intestine. So this little sphere is basically a mini small intestine. And when you look at this image of at the microscopic level, what you have is a circular object, just like the small intestine is. And inside you see little villi. The cool thing about this is that it behaves exactly like the human disease behaves. And we could pick up T cell activation, we could pick up production of those autoantibodies, and we could also pick up intestinal destruction when we treat this with gluten. And what I'm going to show you, hopefully this works, is a video, a time-lapse video of what happens to a celiac organoid when you give it gluten. And live is green, dead or apoptotic cells are red. So this is what this experiment shows. As you see, as time progresses, more of those green live cells are replaced by red cells. So this is a very exciting model. Um, and we hope that it will have some applications for making the diagnosis and to help us stratify the severity of the disease. It may be something that we can do that we may be able to use in patients who are already on a gluten-free diet. We could do the gluten challenge outside of the body. But the other important aspect of this was that in our group discovered that there was a new um, molecule that it wasn't that we didn't know uh, prior to this that was involved in celiac disease, and this is an interleukin 7. And it is something that we are thinking that it's in the planning phase, but the hope would be that a human antibody to IL-7 may be a potential therapy for celiac disease. This already exists, and the goal would be to repurpose this. So stay tuned for that. But also using this organoid system will help us just understand celiac disease at a deeper level, which may have serious implications for other autoimmune disorders that we know very little of. So now let's talk about new tools to diagnose celiac disease. And some of you might have heard of this, but in the course of some clinical trials in the past, it was noticed that a lot of clinical trials require patients to eat gluten because what you have to prove is that the medication you're testing is protecting the subject from developing gluten-induced injury of the small intestine. And some of those trials didn't work. But what we have is a lot of data of what happens to people with celiac disease when they consume gluten. And there was an observation that there's a spike in a chemical, a cytokine that's called interleukin 2, a few hours after ingestion of gluten. So this has been reproduced many times over. And a test, there was a report in the Journal of Gastroenterology a few months ago where the way that the test works is a blood sample is taken from a patient that potentially has celiac disease. And in the laboratory, that blood sample is exposed to gluten in a test tube. And then sometime later, their gluten-specific T cells will become activated and release interleukin 2. And the test and measuring interleukin 2 might give us the ability to make the diagnosis. And in this study, which was not done in the United States, I believe it was in Europe, there was 90% sensitivity and 97% specificity. And this was even in patients who were already on a gluten-free diet. So this is very exciting. And it remains to be clinically validated to see if it's a practical therapy, that a practical diagnostic test that we have, but this could become a very, very helpful tool for us. Now I'm going to tell you about something else that our group is doing here at Stanford under the direction of Chetan Khosla. So this is to remind you about celiac disease pathogenesis because this approach exploits that. So what you have is gluten that are undigested protein gets broken down to peptides, which activate the immune response. And the critical step is that this enzyme, tissue transglutaminase or TG2, modifies gluten to the form that's picked up by the immune cells. And that leads to the T cell activation, the injury to the lining that we talked about. So this approach targets tissue transglutaminase. And what we know about celiac disease is that tissue transglutaminase is active in patients with celiac disease, and it's inactive in people who don't have celiac disease. And this talks about this is a little bit technical, but basically this is what tissue transglutaminase does. It changes one amino acid from a glutamine to a glutamate, which makes the molecule more negatively charged. It makes it much higher, more likely to be picked up by the immune system. So in Dr. Kosal's lab, they engineered a molecule that actually binds to the active form of tissue transglutaminase. And it's linked to a fluorescent molecule that allows us to visualize. So now we are able to pick up tissue transglutaminase activity, and which we think is directly proportional to the degree of mucosal injury. And it's a positive. I'll show you what we've done so far. So we have taken biopsies from patients and treated those biopsies with this molecule. And basically what you have, so on the top, you have a control. That's a person who doesn't have celiac disease. In the second panel, it's somebody who's in remission who has no celiac disease, but well controlled on a gluten-free diet, and it's someone who's active, brand new diagnosis. And what you have here in the bottom panel is the immunofluorescence. So tissue transglutaminase is green, the molecule that was generated in the lab that binds to the active tissue transglutaminase is red, and when they are together, it makes yellow. So what you see is that in a person who doesn't have celiac disease, there's no yellow at all. In someone who's in remission, you see small amounts. But in a patient who has active celiac disease, there's lots of TG2 activity. And so again, this is a potential diagnostic test because it allows us to pick up tissue transglutaminase activity, but more importantly, because of the mechanism of action. At higher doses, we think that can also be a potential therapy in the future. And if we are actually doing a clinical trial testing this right now at Stanford. Um, uh, we have done all of the studies that are mandated by the FDA to be able to do our first in-human study. This is a phase 1A, 1B study looking at safety, and it's underway. If you are interested in participating, there's the information in this QR code. Please let us know. So the last part of my talk is what is the future of medicines for celiac disease? And I'm going to turn to this figure, which by now is probably pretty familiar to you. Here's gluten, here's that single cell layer, the immune system below, in the cascade of the tissue transglutaminase modifying gluten to deaminated gliadin, and that leading to the activation of all the other players that leads in the mucosal injury. Well, it turns out that the drugs that are in trials right now were designed to interfere with different aspects of this pathophysiology. So there are molecules that basically will break down gluten so that it cannot activate the immune response. And so those are enzymatic degradation. Latin glutenase, there was a phase 2 study that wrapped up recently, and it seems to have some efficacy. And the hope is that in the coming year or so, that there will be a phase 3 in making it a little bit more closer to development. There's also molecules that sequester gluten so that it cannot be absorbed. There are probiotics that are being tested that may modify gluten in some way. There's also the same mechanism that I talked to you about that we're exploring in our lab there's been a clinical trial already that shows that blocking tissue transglutaminase activity is helpful in dampening the immune response. And it's currently in clinical trials right now in phase 2, and we'll talk more about that. But there's also drugs that will block specific cytokines like interleukin-15, which is a critical interleukin in the pathogenesis of celiac disease, and other chemicals, as well as drugs that affect the barrier. There's tight junctions, and we know that those tight junctions become a little leakier as the inflammation ensues. And there is something called larazotide, that targets that. So this is the process. It's a very long process. There are phase 1, 2, and 3. Phase 1 tend to be several months, and that is usually proof of concept, trying to figure out safety of the drug in question. Phase 2 is more several months to years and requires a lot more people, and you're looking at efficacy. And then phase 3 is really very large numbers. And that really is what helps us tease out the drug benefits. In the prior phase ones, phase two, we work out a lot of the safety issues if there are any. You could go on the site clinicaltrials.gov and search for celiac disease, and it will tell you all of the clinical trials that are going on right now that are enrolling. I wanted to highlight a few that are active right now. Aspirion is a phase 2A to B. It's the sponsor is Sanofi, and the mechanism of action, there's a chemical an antibody that inhibits something called PD2, and it's been evaluated in malignancy, but it seems to have applications for autoimmune conditions, including immune regulation. It's known to reduce autoimmunity, and it has the potential to induce tolerance. And what inducing tolerance means, and that's actually a mechanism that I didn't tell you about. The other thing is inducing tolerance, which means making the immune system not react to gluten. In a sense, tricking the immune system into just not reacting to gluten, which is what's supposed to happen in the first place. And there are some of those, like number 5 here, that are in clinical trials that are being tested. So the goal here is to see whether amlitelimab is helpful for subjects who have celiac disease but remain symptomatic despite being on a gluten-free diet, and to see whether it could improve intestinal healing and reducing symptoms from accidental gluten exposure. Another trial is, I cannot pronounce these things, ritlecitinib, which is phase 2. The mechanism of action is that it's a small molecule that targets Janus kinase enzymes that are important to propagate inflammation. Also known to involved in immune regulation and reduces autoimmunity. And it's testing, the goal is to test this drug to prevent gut damage and symptoms from gluten exposures in patients who are in remission. And so the idea would be that if there are accidental exposures, that it doesn't cause setbacks or injury. So it would allow you to just be a little bit less anxious about getting exposed to gluten if we could blunt that effect. There is a pretty exciting mechanism which is intended to induce tolerance, again, tricking the immune system not to react. And this is a first-in-human trial of VTP-1000. It's a self-assembling nanoparticle that is injected into the blood, and it serves as a platform to try to re-educate the immune system to stop reacting to gluten. And it's able to deliver multiple gluten peptides derived from wheat, barley, and rye. And the goal is to evaluate the safety and the efficacy of this. And finally, phase 2 trial looking at a molecule that blocks interleukin-15 to prevent gluten-induced injury. Interleukin-15 is also involved in the development of refractory celiac disease, which is the dreaded complication where there's 2 types, refractory type 1 and refractory type 2. And refractory type 2 is the one that thankfully is very, very rare, but carries the risk of a very specific type of lymphoma. and interleukin 15 may be something that will be helpful there. So that's kind of what I put together. I figured that I'd be available to answer any questions, but what I wanted to leave you with is that the future looks very bright. There's a lot of interest in celiac disease, in part because it has so many implications for if we understand celiac disease well, we might be able to understand other autoimmune disorders, but also it affects so many individuals. The treatments, diagnostic tests that we have are not perfect, and there's a very significant drive to change that. So there's a lot of interest and a lot of really smart people that are working on this right now. Thank you so much for your time.